morning students, this is lecture 4 of module 2. In today's lecture, we will be discussing about the biomass properties, how we can enhance some of these properties and what are the challenges in conversion of the biomass into biofuels. So, let us begin with. So, we will discuss about physical properties of biomasses. So, some of the physical properties of biomass affect its pyrolysis and gasification behavior, basically the thermochemical conversion. For example, permeability. So, it is an important factor in pyrolysis. High permeability will allow the pyrolysis gases to be trapped inside in the pores, increasing their residence time in the reaction zone. Thus, it increases the potential for secondary cracking to produce char. The pores in wood are generally oriented longitudinally. As a result, the thermal conductivity and diffusivity in the longitudinal direction are different from those in the lateral direction. This anisotropic behavior of wood can affect its thermochemical conversion. A densification process such as torrefaction can reduce the anisotropic behavior and therefore, change the permeability of a biomass. Hence, permeability is an important property with respect to the pyrolysis. Density is an important design parameter for any biomass conversion system. For a granular biomass, we can define 4 characteristic densities: true density, apparent density, bulk density and biomass growth density. Now, true density is the weight per unit volume occupied by the solid constituent of biomass. So, it is given by total mass of biomass divided by solid volume in biomass. The cell walls constitutes the major solid content of a biomass. For common wood, the density of the cell wall is typically 1530 kg per meter cube and it is constant for most of the wood cells. The measurement of true density of a biomass is as difficult as the measurement of a true solid volume. So, it can either be measured with a pycnometer or may be estimated using ultimate analysis and true density of the constituent elements. Next is apparent density. So, it is based on the apparent or external volume of the biomass. This includes its pore volume or you can say the volume of all the cell cavities. For a regular shaped biomass, mechanical means such as micrometers can be used to measure different sides of a particle to obtain its apparent volume. An alternative is the use of volume displacement in water. The apparent density considers the internal pores of a biomass particle, but not the interstitial volume between the biomass packed together. So, this is the equation for the apparent density. The pore volume of a biomass expressed as a fraction of its total volume is known as its porosity. Apparent density is most commonly used for design calculations, because it is the easiest to measure and it gives the actual volume occupied by a particle in a system. So, you can see the table 1 has given apparent density of some of the wood species. Okay. Then the bulk density. So, bulk density is based on the overall space occupied by an amount or a group of biomass particles. Bulk volume includes interstitial volume between the particles and as such it depends on how the biomass is packed. For example, after pouring the biomass particles into a vessel, if the vessel is tapped, the volume occupied by the particles settles to a lower value. The interstitial volume expressed as a function of the total packed volume is known as bulk porosity. So, this is the bulk density equation. So, to determine the biomass bulk density, we can use standard like the American Society for Testing Materials E87306 standard. So, this process involves pouring the biomass into a standard size box of a particular size, it is given here, from a height of 610 millimeter. The box is then dropped from a height of 150 millimeter 3 times for settlement and refilling. The final weight of the biomass in the box is divided by the box volume, which gives its bulk density. This is how we can measure bulk density of the biomass. The total mass of the biomass may contain the green moisture of a living plant, external moisture collected during storage and moisture inherent in the biomass. So, once the biomass is dried in a standard oven, its mass reduces. Thus, the density can be based on either green or oven dry, depending on whether its weight includes surface moisture or not. The external moisture depends on the degree of wetness of the received biomass. To avoid this issue, we can completely saturate the biomass in deionized water, measure its maximum moisture density and specify its bulk density accordingly. So, there is a relation between these 3 densities as given here, and where rho p is the void fraction or voidage in a biomass particle and rho b is the voidage of particle packing. So, then the next is the biomass growth density, and it is specifically for biomass, not for other materials. So, the term biomass growth density is used in bioresearch industries to express how much biomass is available per unit area of land. So, it is defined as the total amount of above ground living organic matter in trees expressed as oven dry tons per unit area, that is basically the tons per hectare, and includes all organic materials, whether it is leaves, twigs, branches, manbole, bark and the trees. Then we will see some of the thermodynamic properties. When we talk about gasification, which is a thermochemical conversion process, the thermodynamic properties of biomass heavily influence its gasification properties. So, do for pyrolysis also. So, the three important thermodynamic properties are thermal conductivity, specific heat and heat of formation. Now, what is thermal conductivity? Now, biomass particles are subject to heat conduction along and across their fiber, which in turn influences the pyrolysis behavior and or gasification behavior of course. Thus, the thermal conductivity of the biomass is an important parameter in this context. It changes with density and moisture. So, how it changes we will see. So, based on a large number of samples, MacLean in 1941 developed the following correlations, okay, which is adapted from this Kitani and Hall 1989 book, is given in the page number 877. Okay. So, K effective watts per meter Kelvin is specific gravity in bracket 0.2 plus 0.004 into MD plus 0.00238. So, this particular correlation, as you know, correlations are valid for certain range. So, for this particular correlation it is only valid when your MD is greater than 40%. Now, another equation which is given by this. You can see that equation also, I am just not reading it. So, that is valid when MD is less than 40%. So, two equations or correlations are proposed. The first one is when the MD is greater than 40% and the second one is when the MD is less than 30%. So, MD is the moisture percentage of the biomass, okay, on a dry basis. So, unlike metal and other solids, biomass is highly anisotropic. Conductivity also depends on the biomass's moisture content, porosity as well as temperature. Some of these depend on the degree of conversion as the biomass undergoes combustion or gasification. Thunman and Lechner in 2002 wrote the effective thermal conductivity parallel to the direction of wood fiber as a sum of contributions from fibers, moisture and gas in it. It is a good equation which many of us working on the biomass sector they use it. Okay. So, K effective in watts per meter Kelvin, okay, is G Ks plus F Kw plus H into Kg plus Krad for a parallel fiber. Now, where Gx, Fx and Hx are the functions of the cell structure and its dimensionless length, Ks, Kw and Kg are thermal conductivities of the dry solid, that is fiber wall, moisture and gas respectively, and Krad, okay, represents the contribution of radiation to conductivity. It is a very nice or excellent equation which is being adapted universally. Okay. So, we will see few more equations. So, these components are given by the following empirical relations, which are to be used to calculate the directional values of the thermal conductivities. Okay. Here all the thermal conductivities are measured in watts per meter Kelvin. So, Kw is given by this equation minus 0.487 plus 5.887 into 10 rise of minus 3 into T minus 7.39 into 10 power of minus 6 T square. Now, Kg is given by this long equation, Kw is 0.52 in perpendicular direction and Krad, so which is coming from the radiation is 5.33 E of radiation, then here this one sigma D of pore and D pore and Tq. Okay. So, E rad is the emissivity of the pores having diameter D pore and F sigma is the Stefan Boltzmann constant and T is the temperature in Kelvin. The contribution of gas radiation in the pores Krad to conductivity is important only at high temperatures. Now, we will talk about specific heat, another important thermodynamic properties. So, specific heat of biomass is often required for thermodynamic calculations. So, it is an indication of the heat capacity of a substance. Both moisture and temperature affect the specific heat of biomass, but density of wood species do not have much effect on the specific heat. So, the specific heat changes much with temperature. It also depends on to some extent on the type and source of the biomass. So, please look at this particular figure. So, you can see there are three species temperature versus specific heat has been given here. So, the below one, that is the wood char, the red one is the wood bark and the blue one is the wood. So, this figure 1 shows the increase in specific heat of a softwood species with temperature. It also shows that bark of the wood has higher specific heat when it is compared to the other two species, and char produced from this wood has interestingly much lower specific heat. Now, some experimental correlation of specific heat with temperature and moisture content is given as this Ragland et al. Equation, 1.39 plus 0.00036 T for the wood char. Gupta et al. Suggested for the softwood fuel 0.00546 into T minus 0.524, and for the hardwood fuel 0.0038 into 10 power of minus 3 T square plus 0.00598 T minus 0.795. Now, I want to say something about this so called relations. Please note, I do not know whether most of you are aware of the fact or not regarding these correlations. So, let us understand what is the meaning of correlation, why suddenly some particular number of 0.003, some x square, some t square is coming into picture. Now, please understand that any correlation is a equation which is developed by doing certain number, fixed number of experiments. It is all based on the experimental results, that is why they have some specificity or limitation. Like we are showing na MD in the last equation, I told you that this particular two equations, one equation is valid when the moisture content is greater than 40%, another equation is valid when the moisture content is less than 40%. So, that the reason is that this is how the experiments are being done, and this is how the equation has come from different experiments, and mostly they are average values. So, there is a particular way to do it. So, you need to understand that any correlation are experimentally derived equations and has some limitations. So, then let us understand the heat of formation. Heat of formation, also known as the enthalpy of formation, is the enthalpy change when 1 mole of compound is formed at standard state, that is 25 degree centigrade and 1 atmosphere from its constituting elements in their standard state. Now, for example, hydrogen and oxygen are stable in their elemental form. So, their enthalpy of formation is always 0 in elemental form. Now, however, an amount of energy 241.5 kilojoule is released per mole when they combine to form steam. That means hydrogen and oxygen, right. So, the heat of formation of steam is thus minus 2241.5 kilojoules per mole, that is in the gaseous form. So, this amount of energy is taken out of the system and is therefore given a negative sign in the equation to indicate that it is an exothermic reaction. If the compound is formed through multiple steps, the heat of formation is the sum of the enthalpy change in each process step. So, gases like hydrogen, oxygen, nitrogen and chlorine are not compounds, and the heat of formation for them is 0. Values for heat of formation for some of the compounds are given. You can see it later. Water is minus 241, carbon dioxide is minus 393, like there are few words given just for your understanding. Now, there is a small example problem, you can just go through it. So, find the heat of formation of sawdust, the heating value of which is given as 476 kilojoules per mole. Assume its chemical formula to be C H 1.35 O 0.617. Now, stoichiometry has to be written. The conversion of SAW can be written in the simplest term as CHO plus 1.029 oxygen will give C carbon dioxide plus water 0.6575 water minus 476 kilojoules per mole of sawdust is the exothermic reaction. So, heat of reaction you can calculate like this: HF of carbon dioxide plus 0.675 HF of water minus HF of sawdust minus 1.029 HF of oxygen. So, consider the values of HF of heat of formation of carbon dioxide, oxygen and water, and substitute. So, you will get for the heat of reaction for the above combustion reaction is minus 476 it is given. So, you will calculate the heating value to be minus 80.5 kilojoules per mole. So, the heat of reaction is the amount of heat released or absorbed in a chemical reaction with no change in temperature. In the context of combustion reactions, heat of reaction is called the heat of combustion. Delta H comb submits a combination which can be calculated from the heat of formation, as methane plus oxygen gives you 2 water plus carbon dioxide, So, heat of combination will be 2 of this one delta H water plus delta H carbon dioxide minus delta H methane minus delta H oxygen. So, the delta H combo, okay, the heat of combustion of the combination for the entire fuel, so can be defined as the enthalpy change for the combustion reaction when it is a balance. So, fuel plus oxygen will give you water plus carbon dioxide minus heat of reaction. So, the heating value of biomass is the amount of energy biomass releases when it is completely burnt in adequate oxygen. So, it is one of the most important properties of biomass as far as energy conversion is concerned. Compared to most fossil fuels, the heating value of biomass is low, especially on a volume basis, because its density is very low and it is high oxygen containing fuel. Higher heating value, what is higher heating value? This is also very important to understand. So, it is defined as the amount of heat released by the unit mass or volume of fuel initially at 25 degrees centigrade, once it is combusted and the products have returned to a temperature of 25 degrees centigrade. It includes the latent heat of vaporization of water. HHV is also called as the gross calorific value. In North America, the thermal efficiency of a system is usually expressed in terms of HHV. So, it is important to know the HHV of the design fuel. Then there is something called LHV, lower heating value. So, LHV also known as net calorific value. Okay, HHV is gross calorific value and LHV is the net calorific value. So, the lower heating value is defined as the amount of heat released by fully combusting a specified quantity less the heat of vaporization of the water in the combustion product. So, this is the equation you can refer to. So, then the next thermodynamic property, ignition temperature. So, ignition temperature is an important property of any fuel, because the combustion reaction of the fuel becomes self sustaining only above this temperature. So, above only this temperature it will ignite basically. So, in a typical gasifier, a certain amount of combustion is necessary to provide the energy required for drying and pyrolysis and finally, for the endothermic gasification reaction. Exothermic chemical reaction can take place even at room temperature, but the reaction rate being an exponential function of temperature is very slow at lower temperatures. So, when the fuel is heated by some external means, the rate of exothermic reaction increases with a corresponding increase in the heat generation rate. So, above a certain temperature, the rate of heat generation matches or exceeds the rate of heat loss. When this happens, the process becomes self sustaining and that minimum temperature is called the ignition temperature. So, the ignition temperature is generally lower for higher volatile matter content fuel, because biomass particles have a higher volatile matter content than coal. So, usually they have significantly lower ignition temperature. So, they mean that the inherent meaning is that, so biomass this one particles will ignite very fast compared to the coal. So, for example, the wheat straw has a volatile matter of 72% DAF basis. So, DAF is the dry ash free basis. The ignition temperature is 220 degrees centigrade, while the volatile matter of anthracite is only 7.3%, but the ignition temperature is 927 degrees centigrade. Now, we will try to understand how do we will enhance the primary raw materials for the biofuels and what are the different types of techniques that exist, including some of the genetic engineering aspects. Now, the use of plant cell wall as major energy sources would establish a virtuous industrial cycle and thus help mitigate global warming problems, as plant cell walls constitute the natural carbon dioxide sinks. Unfortunately, plant cell walls are extremely resistant to enzymatic degradation and so are difficult to degrade into fermentable sugars. Now, that is the reason why, as it is mentioned here. So, there is a need for the pre-treatment of the biomass. We will also discuss in our subsequent slides today itself. Okay. And due to this recalcitrant nature of the cell wall, a huge amount of energy and effort is required to make it amorphous, thus release the sugars which is responsible for producing your alcohol. Okay. As a result, current dynamic area of research is the transformation and harvesting of plants. So, the cellulose microfibrils, of which could be easily hydrolyzed by cellulose preparations or which could self degrade their cellulose microfibrils by expressing a cocktail of hydrolytic enzymes. On the other hand, now it is clear that such genetic modifications capable of conferring these novel characteristics would be feasible only if the resultant genetically modified plants could achieve adequate public acceptance and were able to strive in natural cropping system. Now, here I wish to tell you something very interesting. So, many of you will be knowing about this genetic modified crops. Okay. You remember few years back in India, there is a lot of hue and cry regarding the genetic or transgenic brinjal. Okay. Public perception about genetic modification till date is not so good. Okay, right. So, they feel that if a particular species is genetically modified and being consumed, okay, by the humans or animals, it may have some bad effect, which I cannot have, I do not give a right straight away answer to that. We need to do more study on that actually. I cannot say whether it is good or bad in this platform. So, but we need to understand one thing is that the public perception is not so good, and acceptability of such genetic crops is actually you need more public awareness, and you need to convince public about whether what is the importance of this, and whether is there any adverse effect if it is being consumed by the humans or animals. Okay. This is first thing. Second is thing, this so called genetically modified species, plants, crops, whatever it is, they must have the capacity of naturally cropping systems. Okay. That is one thing. They should also be able to withstand the usual natural environment as well as the climatic conditions. Okay. So, these are the some of the challenges which are still remain in the development of so called genetic engineering or genetic engineered species, crops or transgenic plants, you can call them. Okay. So, let us understand now and go ahead. So, the current technologies for biofuel production typically involve the pre-treatment of lignocellulose before hydrolysis with cellulose preparations. Cellulase is the enzyme which will degrade the cellulose, okay, to glucose. So, an alternative concept is to either upregulate or downregulate hydrolases by introducing and programming their genes in order to achieve in situ modification of the plant cell 1 polysaccharides. So, we can do in situ modification inside the plant cell itself by doing some genetic modifications by overexpressing either certain genes or certain proteins which is responsible for a particular let us say either increasing the cellulose yield or carbohydrate yield, or making it resistance to certain types of pathogens attack. Okay. There are many things. It is not that genetic engineering is being done only to yield, have a higher yield of the biomass or have higher yield of cellulose. Okay. It is not so. So, in principle the introduction and programming of such genes should not decrease cellulose production levels in plants, otherwise it will have an adverse effect. So, our main aim is not going to be achieved. Okay. So, as such genetic engineering could promote the degradability of cell walls in plants bred for use as biofuels, although the degradative gene products in bacteria and fungi are more effective in digesting polysaccharides than those present in plants. So, plants sometimes produce pathogen related proteins such as antibodies. So, thus it is necessary to create and improve a technical barrier to plant engineering using trans-kingdom genes. I hope you all understand what is genes. Okay. So, you can browse little more about this particular few slides what I am saying, few of the particular words which you may not be aware of, please find it and read it from literature. Okay. So, now we will discuss how we can do this genetic engineering technique using the in febrile modification. Now, cellulose is most abundant biopolymer on the earth. An important characteristics of this biological polymer is that it has a strong tendency to self associate into microfibrils. So, that are not easily hydrolyzed either chemically or biologically, and that accumulate primarily in the walls of the plant cells. Now, since individual strands of cellulose are intrinsically less hydrophilic than other soluble polysaccharides, cellulose crystals tend to form extensive intra and intermolecular hydrogen bonds with complex 3-dimensional structures. In natural crystals, so as for example, cellulose 1, the cellulose strands are parallel and form triclinic cellulose, okay, and monoclinic cellulose in varying proportions depending on their origins. So, the microfibril is drawn with its chain axis as a monoclinic structure corresponding to the native cellulose of higher plants. After strong alkaline denaturation, cellulose 1 forms a thermodynamically more stable structure than that of the cellulose 2 with an antiparallel arrangement of strands. Therefore, cellulose 2 is artificially generated from cellulose 1 by two industrial processes. First is called regeneration and second is called the mercerization. Each microfibril consists of repeated crystalline and non-crystalline regions, each of which might be relatively short. So, almost around 10 to 100 glucosyl residues that it contains. Okay. The microfibrils are too rigid for cellulases to attack both the crystalline and the non-crystalline regions. Lignin, another component, very high class compound. Okay. It may bind to hemicellulose, mainly xylan, thereby associating with cellulose microfibrils and further rigidifying them. Okay. So, in a lignocellulosic biomass, next sometimes I will show you a structure. Cellulose, hemicellulose and lignin are bound together in a very intricate manner. So, thereby making it more rigid to the cellulase attack. Cellulase is the enzyme which we want to use it for degrading the cellulose whatever it is available. Okay. So, that is the reason why we talk about this delignification process. The pre-treatment is all about mostly delignification, not always it is delignification, but mostly it is roughly understood as delignification. That it means removing the lignin or separating lignin from cellulose and hemicellulose. Cellulose is C6 sugar and hemicelluloses are C5 sugars, pentose sugars basically. Okay. Pre-treatment for decomposition of lignocellulose could represent a critical step in the conversion of lignocellulosic biomass, as their function comprises increasing the susceptibility of plant microfibrils to cellulose action. A noteworthy strategy for cellulose hydrolysis is not only to promote decrystallization between the so called 1,4 beta glucans in the crystalline regions, but also to loosen the association between 1,4 beta glucan and hemicellulose in the non crystalline regions. Okay. So, that is very important, this particular sentence is very important. So, the cellulose hydrolysis is not only doing the decrystallization between these 1,4 beta glucans, but it is also doing losing the association between this 1,4 beta glucan as well as other hemicellulases in the non crystalline regions. Okay. So, the use of transglucosylase such as xyloglucan endotransglucosylase, which is known as XCTZ people call it, is yet another potential method for transferring glucosyl residues of 1,4 beta glucan to another chain. Now, this can be exemplified by the action of Baddeley-Z, which catalyzes the transfer of cellulose molecules to xyloglucan and thereby forms a link between cellulose and xyloglucan. It is well known that the removal of lignin results in an increased level of saccharification of plant cell walls, and this method is commonly used to facilitate the process of bioethanol production. Lignin occurs in close association with cellulose microfibrils. So, it is always expected that a decrease in lignin content would in turn increase the accessibility of the cellulose microfibrils to degradative enzymes. Lignin is an important cell wall component in the plants, not only for water transport in xylem, but also for stem straightness and protection against pathogen attacks. So, lignin provides some sort of mechanical support also, you can say that. Therefore, it seems likely that a dramatic reduction of the lignin content in the growing plants would result in a detrimental effects of the plant growth. So, we need to balance it. So, consequently, reducing the lignin content of lignocellulosic biofuel crops appears to be that of practical use. Now, let us understand that if I can give an example, let us understand that if we want to reduce the lignin content of a dedicated energy crop in which the lignin content is already less, let us say miscanthus, switchgrass, elephant grass as such grasses, then it will not, because they are bush type of thing, they are grasses. So, then it will not going to have a much higher effect on the mechanical stability of the plants, these bushes, because they are grasses and bush basically. But having said that, if we are drastically reducing the fibril lignin for the little hardwood or softwood trees, then we need to be careful whether the plant can grow properly and erect and stand on the soil itself, okay, by having a good mechanical stability. So, that is the question basically, that is how the genetic engineering or the engineers, okay, must ensure that there is no adverse effect on the growth of the plant. So, the transgenic populus tremula overexpressing Arabidopsis cellulase, that is cell 1, exhibits longer internodes and longer fiber cells. Remarkably, those characteristics translate into immediate gains in bioconversion productivity. In addition, the enzymatic trimming of amorphous region in the microfibrils leads to the solubilization of some xyloglucan that is intercalated with disturbed paracrystalline domains of the microfibrils. Xyloglucan is a key polysaccharide that is used by the plants to control the assembly of cellulose microfibrils through cross linking. So, therefore, the degradation and reconnection of xyloglucans could induce the modification of cellular polysaccharides in such a way so as to further facilitate industrial saccharification. Since adjacent cellulose microfibrils could be cross linked to xyloglucans, the separation of microfibrils during elongation is thought to require enzymes that solubilize xyloglucan or loosen its binding to microfibrils. An additional enzyme that is an important target for plant genetic engineering is xyloglucanase, which is called ZEG, XEZ, which catalyzes the endohydrolysis of the xyloglucan backbones and exhibit a xyloglucan specific endo-1,4 beta glucanase activity. ZEG is widely distributed in nature, being present not only in plants, but also in fungi and bacteria. So, the overexpression of ZEG in poplar is a plant which we have discussed in our last class. Okay. A dedicated energy can be grown for the dedicated energy crop. So, the overexpression of ZEG in poplar resulted in the cleavage of xyloglucans cross linked with cellulose microfibrils and in an acceleration of stem elongation by losing the cell wall. The overexpression of this enzyme also causes an increase in wall density and cellulose content. So, I will tell you in a crude way what is the meaning of overexpression. Okay. These are genetic engineering terms. So, as I told you, please go back and read little more about certain terms which are not you are not very clear about. So, in this particular class it is very difficult to make you understand each and every bit of the genetic engineering aspects. So, that is not the scope of this course also. So, I will be telling in a nutshell, overexpression means making more copies of the parent protein, okay, or gene, whatever it is. Okay. So, for example, if cellulose formation in wild type poplar is restricted by the entanglement with xyloglucan, the relaxation resulting from the cleavage of cross linking xyloglucans in the modified poplar may accelerate cellulose biosynthesis and deposition. So, the meaning of this particular sentences is that when we overexpress ZEG in the poplar, so it has helped in cleavage of the xyloglucans which is cross linked to the microfibrils, cellulose microfibrils. Okay. So, by these mechanisms the expression of ZEG would promote not only cellulose degradation, but also the production of cellulose in plants. Okay. So, it is a good technique. So, the observation that overexpression of ZEG in poplar results in the acceleration of cellulose degradation by cellulase preparations is consistent with this hypothesis. Now, it is also noteworthy that the reconnection between xyloglucan molecules in the walls can be catalyzed by xyloglucan endrotransglucosylase, which is called the Z, okay, XCT, an enzyme encoded by the gene of XTH gene family. Okay. However, XCH present in plant cell walls has not been well characterized, another enzyme basically class of enzyme. So, there is possibility of some relationship and or interaction might exist between XTH and cellulose synthase gene expressions. It is possible that this mechanism might be leveraged to facilitate biomass processing. More work is currently being done on whatever we have written in this last sentence here. So, French bean cells habituated to grow in the presence of 2,6-dichlorobenzonitrile formed large amounts of soluble beta glucan and evoked the Z activity. Such inhibition of cellulose biosynthesis would apparently not only cause the occurrence of soluble 1,4 beta glucan, but also decrease the plant growth. While the cellulose biosynthesis pathway in plants remain unclear, there is convincing evidence that a relationship exists between cellulose synthase, cellulase, the enzyme and the Z. Okay. One line of research and development to facilitate the implementation of lignocellulosic biomass at a large scale is thus to make the use of this relationship to weaken the cellulose polymers in vivo, which can be achieved by appropriately altering the genetic makeup of biofuel crops. It is hoped that this might be achieved in such a way that the industrial saccharification of lignocellulosic biomass could be performed under optimal economic conditions without affecting the natural ability of these crops to grow in a natural cropping systems. Again, this is what we have already discussed in one of the slide. As I told that everything is so good about the genetic engineering things. Okay. It has to be done in a proper way and proper understanding that if I am decreasing the lignin content, then it should not affect the growth of the plant. This is one of the foremost important thing. Okay. And let us learn what is in planta modification of another genetic engineering technique. A two-pronged strategy is required to improve lignocellulosic crops for optimal biofuel yield. So, the first one, it is necessary to increase the yield of the cellulose production based on the plant mass, while on the other hand it is also necessary to increase the conversion of cellulose into glucose. There are two things. First you increase the yield of cellulose. Okay, by means of the genetic engineering aspect. Second thing is that you increase cellulose, but cellulose is bound in such a intricate fashion with hemicellulose and lignin, and the rigidity is so high, they are crystalline. So, that crystallinity has to be overcome, so that the cellulose can be transformed into glucose, right. So, improvements in the post harvest processing in planta were originally attempted in transgenic tobacco. A transgenic which constitutively produced hyperthermophilic A glucosidase and B glucosidase, two different types of enzymes, okay, from the hyperthermophile Sulfobus sulfatyricus. So, this is one particular unicellular organism from which these two enzymes has been derived. Transgenic plant means genetically modified plant. Okay. So, the transgene glucosidases began to accumulate in the tobacco plant after a certain delay and were inactive at plant growth temperatures. After harvest, however, glucose could be produced from endogenous polysaccharide upon incubation at high temperature. I would like to say that transgenic tobacco is being farmed in many of the western countries, okay, because not for the consumption of the tobacco leaves, but to purify one particular monoclonal antibody, okay, which is present in the transgenic tobacco. Okay. Usually, it is present in very small quantity, about 6 to 7%, not more than that. Okay. Sometimes even more less than that, depending upon the species. So, it is a very high class antibody, it needs to be purified. For that purpose, this transgenic tobacco plants are being cultivated. So, Orbe et al. In 2007 showed that cellulose expressed in rice can effectively convert the cellulose of ammonia fiber explosion pretreated rice and maize biomass into glucose. Now, ammonia fiber explosion pretreated rice. So, ammonia fiber explosion is one of the pretreatment technique. We will read about more pretreatment techniques later in our subsequent lectures. Okay. We will discuss about ammonia fiber explosion also. Okay. So, these authors suggested that such a method of expression could be used as an environmentally friendly technology for the hydrolysis of wasteful rice straw. So, next genetic engineering technique is InCREST-T modification. So, CREST-T means chimeric repressor silencing technology was developed as a novel method to silence the target genes of transcriptional activators in plants. Generally, each transcription factor is, again I am telling you, transcription factor is a protein. And what is transcription? So, transcription means in a nutshell I will tell you transferring one particular information from the DNA to the messenger RNA, mRNA. Okay. So, that process is called transcription. Okay. You can read little more about these terminologies from the literature, so that things will be more clear. In CREST-T, a fused gene encoding a transcriptional activator and a repression domain named as SRDX at the carboxy terminus is expressed as an artificial chimeric repressor. The SRDX is a modified short amphiphilic peptide of 12 amino acids derived from the plant specific transcriptional repressor known as Superman. This strategy has the following advantages over the conventional genetic manipulations such as RNI or gene knockout, particularly in many horticultural plants which have high polyploidy and only limited sequence information. You know, polyploid is something there is something called diploid. What is the meaning of that? So, when a offspring is actually born, it usually carries one sets of chromosomes in their genes from each of the parents, right. In polyploidies, so they will have two sets of genes, basically two from one parent, two from another parent. You can understand in a crude way. So, chimeric repression can dominantly suppress the expression of target genes and induces loss of function phenotype even if the endogenous paralogous genes function redundantly. A plasmid construction is very easy. What is plasmid? Plasmid is a small extra chromosomal DNA, right. It is a extra chromosomal DNA not present in the chromosome itself. So, cloning of the gene encoding the target transcription factor from each plant species is not necessarily required, because the construct of the model plant can be effective in other plant species. To this date, various traits of several floricultural plants have been successfully modified by the CREST technique. Because lignin content and fermentable sugar yields are reversibly correlated the enzymatic saccharification rate of plants without secondary walls in their stem may be higher than that of the plants enriched in the secondary walls. Some additional modifications may be required to utilize plants lacking secondary walls, because their total amount of cellulose is decreased, thus preventing the plants from standing erect and making them very fragile. Further analysis of each plant species is required to evaluate whether these disadvantages could be compensated by the positive attributes exhibited by plants that lack secondary walls. It is particularly worth noting that a reduced lignin content in secondary walls improves the glucose yield. That is what we have already understood. Since lignin and cellulose, the major components of secondary walls, are polymers of completely different molecular classes and result from unrelated biosynthetic mechanisms, each component of the cell wall is likely to be independently regulated by different transcription factors downstream the NST genes. So, you please I am leaving it as it is, you please read this later on. If you have any query, please ask me. This little more detail about the genetic engineering aspect, though is not so much important for this course, but I felt that I will basically write it. So, you can later on read it. So, I am just moving ahead with the other material. So, this also the same thing. I am just leaving it to you for to read. In case you have any query, please feel free to write to me. I will definitely happy to address those. Now, we will try to understand what are the challenges in conversion of biomass to biofuels. There are only few challenges, but there are that needs to be addressed suitably, so that we will have no problem in the conversion basically, right. The first one is the moisture content. We have discussed it in a nutshell earlier. So, biomass materials with high moisture content is not suitable feedstock for conventional thermochemical conversional technologies such as gasification pyrolysis. High moisture can reduce the effectiveness of conversion processes. Moisture in raw biomass materials is also undesired, because fuel wood produced from these materials can contain more moisture. The fuels which have high moisture contents cannot be burned easily. Some part of the energy in the fuel are always consumed for the vaporization of water, which is present in the fuel. In order to maximize the heating value of the fuel produced from these materials, the moisture content biomass should be always less than 20%, as less as it is possible. Drying the materials before being used in the conversion process is not preferable because of high cost, because it is a energy intensive process, drying. On the other hand, some biomass conversion processes use biomass with high moisture content. The first one is hydrothermal conversion process. This is a beautiful technology. It is currently being this one adapted in many industrial practices. So, it is in this particular technology, in a high pressure system, high pressure high temperature system, you are going to convert the high moisture content feedstock. It can be anything, any biomass or anything, to crude oil, basically bio crude. And in certain biological processes such as alcohol production from carbohydrates by biomass, high moisture content do not have any do not create any problem. So, in these processes moisture in the biomass plays an important role in the conversion, either as a major reactant or as a reaction environment. For example, high biomoisture content in biomass causes biological degradation, mold formation and losses in the organic content during storage that could reduce the yield of the fuel wood from these materials. Storing biomass at less than 10% can extend the conservation time of the materials and reduce major losses, that means losses of the sugars during the storage period. The drawbacks of high moisture content can be mostly solved by compressing the biomass material for more uniform properties, and that process is called densification. So, you must have heard about densification of biomass. So, increasing bulk density of biomass materials by densification reduces transportation cost and storage volume. However, this process add on extra cost. Densification is an added process basically. So, any added process there is a cost to it. So, the overall cost increases. Then density, bulk density of lignocellulosic biomass materials is generally low. This creates difficulties to handle such large quantities of feedstocks and increases the transportation and storage cost. The bulk density of biomass should be between 190 to 240 kg per meter cube for efficient transport in various sizes of trucks with approximately 25 ton loads. The size, shape, moisture content, particle density and surface characteristics are the factors affecting the bulk density of a material. The challenge for low density and different size and shapes of biomass can be overcome by densification process. So, biomass in densification process, biomass materials are mechanically compressed to increase their density and convert them into uniform shapes and sizes. You can see how these are been converted into particular shapes. These are powder, these are some sort of briquettes, these are some sort of rolls. You can briquetting them, pelletizing them, cubing them. So, then density of biomass material can be increased tenfold depending upon the biomass type, moisture content and processing condition. The costs for handling, transportation and storage resulted densified materials can be considerably reduced. Now, because of uniform size and shape, the materials can be easily handled. So, the next is complexity and diversity. Lignocellulosic biomass materials is mainly composed of three components: lignin, cellulose and hemicellulose. These polymers are organized in the complex non-uniform three-dimensional structures and each one has different polymerization degrees. Polymerization degree and structures of these biopolymers can vary among the biomass species. Cellulose is a linear structure composed of beta 1,4 linked glucose subunits. Cellulose molecules determine the cell wall framework. The inter and intra-chain hydrogen bonding in the structure makes the cellulose to be crystalline, and this portion of cellulose does not hydrolyze easily compared to the amorphous cellulose structure, and that is what we have understood during the genetic modification steps that we have discussed. How the cellulose can be available or more amenable to degradation, either by removing lignin, decrease the lignin content or over expressing certain cellulases enzymes basically, so that whatever we want that will be fulfilled. Hemicellulose has a random and amorphous structure which is composed of several heteropolymers such as xylan, galactomannan, arabinoxylan, glucomannan and xyloglucan. Phenyl propanoid monomeric units in the lignin polymer are identified as p-hydroxyphenyl, guacyl and syringil units. Composition of lignin, cellulose and hemicellulose in biomass materials significantly differ among biomass species. For instance, some biomass materials such as hardwoods contain more cellulose in their structures, while others such as straws have more hemicellulose. Hemicellulose fractions of softwoods mainly have D-mannose derived structures such as galactoglucomannans, while hemicelluloses in hardwoods have D-xylose derived structures. Now, this diversity among biomass material can significantly affect the conversion process for production of biofuel and other useful products from the biomass materials. So, the next is recalcitrance and dissolution difficulties. Success of using lignocellulosic biomass for biofuels and other useful chemical productions depends largely upon the physical and chemical properties of the biomass, on pretreatment methods and optimization of the processing conditions. The compositional changes in plant cell wall and the differences in ultrastructure greatly influence the pretreatment and hydrolysis efficiency of the biomass. Hydrolysis is a chemical reaction that releases sugars from biomass structures. Biomass dissolution involves both physical, chemical and or thermochemical treatment process. We will read more about these techniques later on in our subsequent lectures. So, things will be more clear than that time. So, the crystallinity of cellulose, hydrophobicity of lignin, embedding the cellulose in lignin hemicellulose matrix, difficulties in cleavage of some linkages, as for example, hydrogen bonding, either linkages between phenyl propane units. So, all these things make biomass materials resistant to hydrolysis. Hydrolysates from biomass can be used for producing a wide range of value added products, including biofuels. It can be ethanol, hydrogen, butanol, any such things. Industrially important chemicals, as for example, some of the solvents and food products, sugar and sugar alcohols, etc. Significant existing challenges for hydrolysis of lignocellulosic biomaterials include the following. So, first is that existing hydrolysis methods are expensive and time consuming, most of them are not environmental friendly. Second is that additional steps are required, just like your pretreatment, neutralization, etc. Then third is released carbohydrates decompose in harsh hydrolysis condition, which is prevalent during the hydrolysis process. So, these are some of the challenges needs to be tackled. The major hydrolysis processes typically used for solubilization of biomass require either use of toxic, corrosive and hazardous chemicals, as for examples mostly it is acids, sometimes alkali also, and longer retention time, as for example, during enzymatic hydrolysis. So, which collectively make the process environmentally unsafe and or expensive. That is the need why there is a huge work right now going on across the globe to develop different pretreatment techniques. Basically, different pretreatment techniques, I would rather say that efficient and sustainable pretreatment technique, in which the yield will be more. The technique should be environmentally benign, it should be a green approach. So, huge work is going on. There are developments of hybrid techniques. We will discuss something. Hybrid means basically combining more than one unit operations together. Because in one single unit operation you may not achieve the yield which you are looking for. So, you combine two processes, but having said all these things, two things has to be three things we should note with respect to the pretreatment. First is that it should be a low cost technique and it should be done at a speed at a very faster rate. So, time is directly related to money in industry. Second, it should result in a higher yield of the cellulose. Third is that it should be a greener process, right. So, concentrated acid hydrolysis has been applied, but the problem with concentrated acid hydrolysis are several. So, though they provide higher conversion, but there are environmental concerns, corrosion and so many other things. So, due to all these things, some of these are listed here, please refer later on. Since almost 2 decades, researchers have focused their attention to dilute acid pretreatment rather than concentrated acid pretreatment. At dilute acid pretreatment, you will see hundreds and hundreds of literature reported by various researchers who have worked with so many different types of species and studied the pretreatment using the dilute acid method. So, we will of course discuss more about that. So, subcritical water is an alternative way to hydrolyze lignocellulosic biomass, but please note that when you talk about subcritical, supercritical, the reactor in which you are going to achieve it, the initial investment is very high and you are going to again use higher energy to achieve that. Now, this table will make you understand about certain methods, breakdown methods, pretreatment methods, alkali acidic, enzymatic, subcritical water, and their various advantages and disadvantages. So, please refer to it later on. So, the presence of a weak acid is subcritical water media can also improve hydrolysis of biomass materials. The use of carbon dioxide is a pressurizing gas also caused formation of carbonic acid that plays catalytic role in effective solubilization of biomass. Some studies have indicated that the addition of small amounts of hydrogen peroxide can enhance lignin removal. The differences in the content and composition of resulted hydrolysates can change the yield of the biofuel, that is another concern again. So, for maximum usability, biomass components in hydrolysates should be further broken down into smaller molecular weight components with a suitable method. So, there are other challenges also, we will just quickly go through it. So, although energy demands are continuous, biomass materials are seasonal. So, some biomass feedstocks have advantages in terms of production, harvesting, storage and transportation compared to others. So, perennial energy crops such as switchgrass and miscanthus do not need to be replanted each year, and they do not require special care and high maintenance to grow. In other hand, agricultural biomass residues, whether it is a corn stubble, wheat straw, rice husk, crop peels, pulps etc., are promising low cost feedstocks, since they do not need additional land for biomass growth, and the land used for agricultural belongs to this type of biomass materials. However, high cost of their harvesting and transportation limit their use. In addition to the advantages and disadvantages listed above, different sources of biomass feedstocks do not have the same composition, uniform size and shape etc., that considerably affect the efficiency of the conversion process for a specific product. So, there are so many things that needs to be taken care while you go and design for a particular conversion technology. Therefore, biomass feedstocks for a biorefinery needs to be standardized. This is the ultimate thing and has to be done. So, with this I wind up. Thank you very much. In the next class, that will be module 3, we will start discussing on biorefinery. We will try to understand what is the concept of biorefinery, though in a nutshell I have covered it in the introduction class, and what are the types of biorefinery. So, thank you very much. If you have any query, please drop a mail to me at kmohanty@iitg.ac.in, or please drop your query in the SWAYAM portal. Thank you.